Good day. Today I'll be speaking to you about native fish recovery, management and their habitat associations following the eradication of invasive predatory fish from the Rondegat River in South Africa. To give you some of my background, my name is Casey Broom. I have a master's degree in zoology from Stellenbosch University, where I focused on the thermal ecophysiology and animal personality traits of clinate fishes in the marine intertidal zone. My passion from a young age has been for freshwater systems, particularly with regards to fish behavior and ecology. To this end, I'm currently working on my PhD, where I'm looking at the ecology of native freshwater fishes of the Olifants River tributaries in the Cape Floristic Ecoregion. I'm supervised by Olaf Feil and Josie South at the South African Institute for Aquatic Biodiversity. South African freshwater ecosystems are imperiled by multiple threats. Among these, anthropogenic effects such as invasive species and habitat degradation are the most notable. In the Cape Floristic Ecoregion, a hotspot of species endemism in the southwestern part of the country, most systems are completely invaded and most native species are imperiled by predation and habitat loss. A system of primary concern is the Olifantsdoorn water management area, as the Olifants River of the Western Cape is home to a relatively high diversity of endemic fishes, most of which are classified as vulnerable or endangered. The Rondegat River, a tributary of the Olifants and shown on the map to your right, is a focal conservation system which is a microcosm of the situation of the greater ecoregion. In response to this dire conservation situation, the Rondegat River served as the site for a pilot collaborative alien fish eradication project using chemical methods in South Africa. The Rondegat is characterized by pristine headwaters with progression into lower reaches, where the human impacts of water abstraction, damming, and agricultural activities affect previously invaded lower reaches. The Rondegat was intended to be the first of 4 invasive fish eradication and restoration projects, having ideal characteristics for rotenone treatment, including uninvaded habitat for native fishes to recolonize from, physical barriers to reinvasion, and low angling importance, and thus low impact on human recreational or subsistence activities. It was also recognized that, for an intervention of this scope, an effective low impact, low cost monitoring effort would be needed over an extended period of time in order to properly assess the merits of this conservation action. In the Rondegat River, the focal species are the cyprinid assemblage. First is the Clanwilliam redfin, classified as near threatened. It has a terminal mouth with 2 pairs of barbels feeding on suspended invertebrates and detritus, and is widely distributed in the western CFE. The fiery redfin is endangered with only 5 known subpopulations. It is characterized by a subterminal mouth and 1 pair of barbels. It appears to be a substrate forager and attains smaller sizes than the Clanwilliam redfin. The Clanwilliam yellowfish is near threatened and is regarded as a flagship for conservation as the largest species in the CFE. It is found primarily in the lower reaches in large oversummering pools, feeding on the substrate of the subterminal mouth, and is a migrant spawner. A pre-treatment survey in 2011 revealed that where invasive smallmouth bass were present, native minnow species were absent and thus locally extinct in the invaded zone. Clanwilliam yellowfish were able to persist as non-recruiting adults. Rotenone treatment was carried out in 2012 with a follow-up treatment adhering to established protocol in 2013, when smallmouth bass were found to still be present. Surveys after the second treatment showed that invasive species had been successfully eradicated from the treatment zone. The recovery of the native fish community was monitored by means of snorkel and camera surveys from 2012 to 2017. There was an initial colonization surge within the first year of treatment, with around 3,000 young of year found in the treatment reach. By 2015, the population in the treatment reach started to resemble that of the control reach, and by 2017, fish density was similar between treatment and control reaches. The Rondegat monitoring program now had 5 years of data to investigate population recovery trends. Recently, Castaneda et al. Looked at the recovery of the system using multi-season occupancy modeling to investigate these trends and determine the annual probability of colonization and extinction. This is a small part of her results, focusing on colonization and local extinction probability. The black arrows show the timing of rotenone treatments. All species follow a similar trend of low colonization probability following the second treatment, with a particularly notable dip in 2015. So they suggested that the dip in colonization probability was a result of both intrinsic and extrinsic factors. An extrinsic factor identified was a very low rainfall year in 2015. It was suggested that this low rainfall resulted in decreased habitat connectivity and increased competition in the remaining habitat, leading to higher densities and encouraging dispersal and an increase in colonization probability after this year. Model averaging estimates taken from multiple candidate occupancy models indicated that overall, the near-threatened Clanwilliam redfin and yellowfish are at population equilibrium, but the endangered fiery redfin is still recovering. My own work on monitoring this fish community commenced in 2018, where I deployed cameras for underwater video analysis in 51 sites across 21 kilometers of river, recording physicochemical and habitat characteristics at each site. The video footage was analyzed in event measure where I established estimated relative abundance or max-N, which is the maximum number of fish per species in a single frame over 15 minutes of footage. This gives a conservative estimate of fish abundance, but it lacks a spatial dimension and thus cannot estimate density. This is the longitudinal profile of the river showing the 2018 monitoring sites, elevation over distance, and the relief of the area. Sites were approximately 200 meters apart and aimed to cover adjacent pools, runs, and riffle habitats. The relative abundances of the three species in April 2018 are shown here. The abundance distribution suggests that the Clanwilliam yellowfish in yellow was fairly abundant in aggregations in oversummering pools in the lower reaches, consistent with previous monitoring. Clanwilliam redfin in grey was found in good numbers across the system, coexisting with both other species. Fiery redfin in red was the least abundant and only in larger numbers in the extreme upper reaches. In October 2018, things changed quite drastically. The large aggregations of Clanwilliam yellowfish seem to have dispersed into smaller pockets across more of the river, and both of the redfin species are more spread out and in lower abundance than they were in April. So to summarize and quantify this overview, we find that relative abundances did indeed decrease between April and October, while detection rates increased from 61% in April to 86% in October. However, despite these seasonal fluctuations, you can see that abundances are still comparable to control region mean values in the post-treatment surveys. Monitoring efforts are still ongoing through 2020. In trying to explain the patterns observed in 2018, a likely possibility is that a major fire event in February 2018 has led to excessive sand deposition as a result of increased erosion from vegetation loss, leading to these large sand flows. In the top left, going from left to right, you can see a site that was a large, deep, rocky section that had healthy numbers of all three species in April, and which was transformed into a shallow sandy stream by October. These two photos were taken from the same spot, while the third shows how the course of the river was changed by the sand slug. On the right, I show the general effect on the banks of the river. During the April 2018 camera survey, you can see these fiery redfins swimming in water that was still inundated with ash from recent rains 2 months after the fire. This has likely forced the dispersal of fishes into new pockets of refugia, with a major reduction in feeding and spawning habitat as the sand encroached downstream by October 2018. So to look into the habitat associations of the three species, I used non-metric multidimensional scaling analysis to visualize the patterns of abundance. The three species separated out quite strongly with regards to their primary habitat types, each represented by a different color shaded circle here. For example, the fiery redfin showed a strong association with vegetated substrates. Mixed effects models also indicated it has a clear headwater stream preference from the negative association with distance from the uppermost site, and negative associations with physicochemical variables such as electrical conductivity and turbidity. On the left, I've shown some of the footage from the underwater cameras, showcasing the diversity of habitat that these fish are occupying. Looking at the general trends from these analyses, the Clanwilliam yellowfish appears to be a main stem migrant and is associated with sandy substrates and typical lower reach characteristics. For the Clanwilliam redfin, weaker habitat associations in any direction support the ubiquitous distribution of the species, which appears to be a classic generalist and may be quite plastic or adaptable. These findings support the previously mentioned occupancy modeling and suggest that two of the species are likely to be somewhat resilient to disturbance, but the fiery redfin is more vulnerable to anthropogenic impacts. Another way to determine if the community has recovered to pre-treatment population levels is to look at size frequency structure. The plots here show initial survey's findings of fork length in the two redfin species. And here's the structure for the Clanwilliam yellowfish, where you can see the species was generally much larger where it persisted with invasive bass and seems to have had very low levels of recruitment success. By 2015, this is what the size structure looked like compared to the control region measurements at the time of treatment. It seems that by 2015, things had mostly returned to control region structure and recruitment was now succeeding. Unfortunately, getting these length frequency data usually requires stressful and high-effort fish capture techniques. In order to monitor the size frequencies and population health in 2019, I use stereo remote underwater video systems. These systems allow us to get accurate fish length measurements without having to stress specimens during handling. I deployed these systems to the same sites as in 2018 for comparative purposes. Some caveats with the system are that it requires careful calibration and is quite a time-consuming method. The preliminary stereo RUV length data were similar to that found in the control reach in previous surveys using traditional methods, with similar fork length structure in the two redfin minnow species. However, high turbidity from abnormal sediment deposition following the 2018 fires made these deployments less representative of the full sample. Thus, some of the major sites for Clanwilliam yellowfish in particular were too turbid to be recorded. Future stereo camera deployments are hopefully still to be carried out within 2020. In conclusion, the Rondegat fish community appears to have mostly recovered from an invasion and a major conservation intervention. Currently, habitat degradation linked to anthropogenic activity is the greatest threat. These threats include damming, abnormal fire events, drought, and their resulting cascading effects. Fire prevention and limiting human activities in the lower reaches are recommended, as well as maintaining the relationship with landowners and continued support for invasion and impact awareness. Finally, this project should be used to inform future invasion interventions in the Cape Floristic Ecoregion. I would just like to acknowledge the many institutions and people that have made the project prior to my contributions and who have helped me with my part. This includes my host institutions for funding and support, the BioLab and SAIAB platforms, as well as organizations such as the Freshwater Research Center and Cape Nature, and the various landowners in the region who have helped me along the way. And thank you very much for your time. Please feel free to leave questions for me in the provided space, and I look forward to answering them. Thank you.